Okay, we got a little interruption there. Every time you get a phone call, it kicks you off of this. So hopefully you guys will appreciate these reviews because they take hours to upload and they're not always the easiest things to do. And I'm probably lecturing too much for a review. We were kicked off here, so that we'll put this on as a part 2. We'll have a part 1 and part 2. But again, arteries, 3 layers. Tunica media is highly developed because it's carrying blood under higher pressure in the veins. Capillaries are 1 layer only. If you had my 137 class, that was in the tissue study. Simple squamous epithelium, we gave you 2 locations, the alveoli of the lungs and capillaries, all capillaries. This is because we want diffusion. That was what the function was. So endothelium only, that means simple squamous epithelium. That's something good to know. Let's go on here. Those are veins. Okay, we're going to do something here real quick. I don't ever put numbers on the test, but this little schematic here kind of describes that, this all has to do with where plasma is at. On the arterial side, plasma is still in the vessel. So that means the outward force of the plasma is greater than it is on the outside of the vein, what we call the interstitial space, or interstitial space, basically. So if plasma is still in here, it wants to get out, and because the outward force is greater, it's going to go out. Once it goes out, there's an immediate drop in pressure, because we transition right to the venous side. So all the plasma that's out here now, that left the building, now creates more of an inward pressure. And that inward pressure will cause the plasma to be taken right back in. This is not an open-ended system. This is closed, and plasma goes in and out, either with nutrients, oxygen on the way out, and then waste products, carbon dioxide on the way in. So that's why it comes in. All that plasma doesn't get back in. That's where the lymphatic system takes over, and we'll talk about that in chapter 16. Again, veins, thinner walls and arteries because they are carrying blood under lower pressure, but they do have valves that help return blood. I'm going to talk about that here in just a minute. In fact, let's do it right here. Sorry, I'm going forward and backward. There's 4 factors that aid in the return of venous blood back to the heart. One of those is valves. And you can see this here. It's like the elevator you get on. You have contraction of smooth muscle, skeletal muscle, which forces blood up. But say this is down in your foot. What's it going to want to do? It's going to want to go right back down because of gravity. But as it does, it causes these valves to close, and it takes it up systematically. Smooth muscle is the second one. Smooth muscle in the vein itself, in the tunica media. Thirdly, skeletal muscle. When you're contracting your calf, for example, in your lower leg, it's helping push that blood up. And the fourth one is respiratory movements. Actually breathing will increase the pressure in the abdominopelvic cavity and below and aid a little bit in that return. Those of you that are going to be nurses, I always tell the nurses in person this, you're going to be walking on floors like I'm standing on now, concrete and tile. Many, many hours a day. You might be lifting people. You should never lift someone by yourself. Get help. Breathe. Breathe when you lift somebody. Don't hold your breath. This is one of the worst things that can cause back pressure, varicose veins, and you have to have, you have great problems with that. So know those 4 factors. Blood pressure, let's go on one slide here. We're going to change one word here. I want you to know the definition, and I'm going to change this a little bit. Systolic pressure is the maximum pressure exerted against the walls of the blood vessels during contraction. Ventricular contraction. Just contraction is the key word there. Diastolic, we're going to change this word minimum to maximum. I think they're trying to make a point here, but I think it's still the maximum pressure you're going to see when you're taking a blood pressure. So diastolic pressure is the maximum pressure exerted against the walls of the blood vessel when the heart or the ventricles are relaxing. Again, relaxation, contractions, the key words. Contraction means systole. Relaxation, diastole. We covered that earlier. These are pulse points. Why do we have so many pulse points? Well, what if they say, Nurse Smith, go down into the ER and take this person's pulse, radial pulse, and they don't have any arms? And I'm not saying that to be funny, that could happen. But you're not going to find a radial pulse on someone with no arms. So you have other areas to check the pulse. This also gets into things like differential diagnosis, which I won't go into, but just real quickly. Someone could have a decreased dorsal pedis artery pulse because they have a blockage in the vascular system. Or they can have a decreased pulse because the nerve is being trapped up here in the spine somewhere by a herniated disc, and it's not telling the blood vessel to dilate. So those are two different things causing the same problem. What I want you to do on your second lab exam, and I'm just going to say it here, I'll probably remind you when I do the review. This could be on here, and it'll be asking you about where you primarily take a pulse, what's the most common place, which is the radial artery, where do you take it, what are you listening to when you're taking a blood pressure, the brachial arteries, something simple like that. You keep that in mind for your second lab exam. This is a great flow chart. I don't think there's anything in there specifically you need to know test-wise that, other than what I told you about the definition of blood pressure. There is one formula on the test. You do not have to measure anything. You don't have to calculate anything, rather. But CO equals SV times HR. This stands for cardiac output equals stroke volume times heart rate. What does that mean? Stroke volume is how much blood am I pushing out of the ventricles. Heart rate, how fast am I doing this? So this gives you your overall cardiac output, and cardiac output should be about 5 liters per minute. In other words, your entire blood volume should circulate through in a minute's time. So this is how we get it. But if you have an increase in heart rate, your stroke volume is going to go down because it can't fill those chambers fast enough. Vice versa, if you have an increased stroke volume, your heart rate is probably going to go down because it can't push that much blood out at one time. Both can affect cardiac output, but they also will respond to the other. If my heart is beating too fast, it may be beating too fast because my heart's not filling quick enough. Maybe there's something wrong with the cardiac conduction system, that electrical system, and vice versa. But just know that as a theoretic formula. There's another one here that you can never read this. It says blood pressure is equal to cardiac output times peripheral resistance. What is peripheral resistance? How big is the diameter of my blood vessels in my entire body? They cannot measure that, but it's a good theoretic formula. 5 milliliters a minute times how much it's being resisted. That's what determines your blood pressure. That will not probably be on the test, but just worth mentioning. These are a good read to give you a little walkthrough on the parasympathetic and sympathetic control of the heart. You can read those along with what I told you earlier about what happens if your blood pressure goes low or high. We already talked about venous blood flow, the 4 factors. That's kind of talking about that. And I think that is it. You have a lot of pictures at the end of this, and I probably should have taken them out of the PowerPoints, but all those are blood vessel drawings. So that is the end of that chapter. Let's quickly go through chapter 17. This is the last one. These are new reviews. There's going to be a couple old reviews posted for lecture exam 2 and 3. I did not have one for exam 1 because we had to go online abruptly due to COVID. So some of these are new, some of these are older, and I'm probably talking way too much. My reviews are usually about 15 or 20 minutes on 3 chapters in person. So I'm probably giving you a lot more information. Okay, the digestive system. I'm going to be very quick on this one because I've spent too much time. This slide actually contains a lot of things that, if you get pressed for time, you can look at this and find some of the basic functions of each part. And basically, that's what the digestive system is. We're going to talk about an area or an organ, and I'm going to give you a function for the most part. So let's get on this quickly. A couple things here, a couple review items that could be on the test. What type of epithelium is found in the digestive tract? Simple columnar epithelium. They show that on the top of this, what we call a villi. You see these villi sticking up. You may remember the microvilli that are associated with these. They're for absorption. That was the function. We have so much surface area in here because of this. This is all area to be absorbing food. And if you look into this particular villi here, you're going to see a capillary network and a lymphatic capillary that will hopefully attract any bacteria, virus, anything nasty in there will go into the lymphatic system, because that is a function of it, is also to fight disease or to fight infection. Otherwise, the food, the nutrients will get into the bloodstream and be carried to the body. Let's go on. Go on. The movement through the digestive system. I don't know that they put the word on here at all, but if you look at the middle and the last picture, there is a movement called Peristalsis. And that is the rhythmic contraction, it's an S on the end. That is the rhythmic contraction of that tube, and that begins in the esophagus. If you remember from last semester, the vagus nerve was the primary parasympathetic supply to the whole body. This is one part it does. It causes that smooth muscle to start contracting. I've had patients that couldn't swallow, and they couldn't swallow because their vagus nerve, something was wrong with it, and it wasn't causing the esophagus to push that food efficiently to their stomach, which can cause you to feel like you get food stuck in your esophagus. So again, this is the rhythmic contraction. The reason that happens, let me go back a couple slides here. If you look at the alimentary canal, you have two layers of muscle. One is circular, one is longitudinal. So they are contracting tangentially to each other, causing this squeezing. If you've ever taken dough and kneaded it, K-N-E, I believe A-D-E, kneaded dough, you know what I'm talking about. You squeeze that, and that's how that does. Due to the conflicting tangents on the muscle. Okay, you should understand if you have sympathetic versus parasympathetic stimulation to digestion, if I'm running from a line, I do not need to be digesting food or defecating. So that is the fight or flight sympathetic. So it is actually going to inhibit the digestive system, where the parasympathetic will keep that acting properly, stimulated. Okay, the mouth, I'm going to be very efficient here because I've taken too much time already, and this is going to take 16 hours to load probably. The mouth is where chemical digestion of food begins. We know that mechanical digestion through mastication, which is chewing, this is breaking up of the food, but chemical digestion begins here because you have salivary amylase, which will break down, which is an enzyme that will break down carbohydrates, simple sugars. I'm not going to test you on the teeth and the gums. We just know that these all have a function in digestion, obviously to gnash up the food. The lips, some people bite things off their lips, but more than anything, the cheeks and the lips create basically a barrier for food not to escape. So it's going to direct it along with the tongue to the back of your throat so you can swallow. Again, the tongue, we're probably not going to put that on there as a function or anything, but it does direct the bolus of food to the posterior of your throat to swallow. We have a hard palate. We talked about in 137 with the palatine processes of the maxilla and the palatine bones. But the soft palate is what we're looking at more here in the uvula. That is going to rise and close off the nasal cavity when you swallow. If you're one of these people that has a friend that you can get laughing while they're drinking something, sometimes those people, that stuff will shoot out their nose because they get it up there before the uvula can close that off. They have a lot of different pictures on this. The teeth for the test, I want you to know how many primary or deciduous teeth are, how many secondary teeth there are permanent. Let's go on. We're not going to learn the parts of the tooth. Used to do that in the lab. The salivary glands, there are 3 of these. These will be on your lab test. This diagram here actually at the top. But the parotid gland is the only one that really has a digestive function per se, as far as breaking down of food, and that's because it is what is secreting the amylase. The other 2 help with digestion by secreting mucus and serous fluid. But enough said on that. All right, the pharynx is a word for the throat. In 137, I do not make students learn the parts of the pharynx. I will mention them, so if you're reading a scholarly article, you'll know what they mean. But we have 3 parts to the pharynx: naso, oro, and laryngopharynx. The nasopharynx, we have everybody knows where your nostril is. We actually call those the external nares, N-A-R-E-S. There is a opening here that's called the internal narrows. If you can see my pointer, I'll just get up and do it here, in case you can't see the pointer. External narrows or nostril. The internal narrows is this opening coming out of the nasal cavity. So from the internal narrows to the uvula is the nasopharynx. From the uvula to the epiglottis, that remember we had elastic cartilage in that in our tissue study in 137. That is your oropharynx, and then below the epiglottis is the laryngopharynx. So if you're reading an article, that might tell you where these are at. I'm not testing you on those areas. They have a lot of pictures in the text on it. They must really like them. I'm also not going to test you on the swallowing mechanism, and I don't want to make light of this because, again, if you have swallowing problems or vagus nerve problems, this can be a very unhandy thing. But just realize that peristalsis is going to take over once that food is in the esophagus if everything's functioning properly. Okay, the esophagus simply connects the mouth to the stomach. I'm going to speed up here a little bit. Again, the esophagus lies behind the trachea. We see the trachea on this cadaver over here on the right-hand picture. See my pointer coming up and down, and then the esophagus in blue behind it. The stomach. The stomach is not your digestive organ. There it is. That's the urban legend that the stomach digests your food. It does not. The small intestine does. However, the stomach is the primary site for chemical digestion of protein. It begins the breakdown of protein. So write that down. It's not in the notes. The stomach actually mixes food. It has 3 layers of muscle rather than 2. This is absolutely stupidity in your notes saying there may be a 3rd layer of fibers. Well, we do things called dissections in cadavers, and we know that people have 3 layers in the stomach. So there are 3 layers. You have a circular, longitudinal, and then a transverse. And these are going to, just think of the movement there. If you've got 3 layers of muscles tangential to each other, it's going to create havoc. And that's where the mixing comes from. Parts of the stomach we'll cover in a lab review. Let's go on here. All right, gastric secretion. Down in the pit of the stomach, you have cells that secrete certain products. Hydrochloric acid is secreted in your stomach. That's why, not to make fun of anybody, but it always tickles me when people say, well, I secrete too much acid. Well, you should. That's your stomach being able to break down protein. But what cells do that? The chief cell is going to secrete pepsinogen here on the first line. Pepsinogen, in the presence of hydrochloric acid, will become pepsin. And pepsin is an enzyme that breaks down protein. This is why you need acidity in your stomach to form pepsin, which is an enzyme that breaks down protein. Hydrochloric acid is produced from the parietal cells, so know that. We have mucus cells in there. Without mucus cells, the secretions of the stomach will digest the stomach. And intrinsic factor, I don't want to make light of this, I probably won't test you on this, but you need intrinsic factor to absorb B12. And B12 is a very important nutrient for nervous system transmission, and that's also produced by parietal cells. So down in these pits, there's all kinds of things being secreted. Let's go on here. This is a very important little video. I can't play it. But basically what this tells you is that hydrochloric acid in your stomach is produced as a byproduct of carbon dioxide in your lungs. And I'll probably mention that in the respiratory review, and who knows, I don't test you on it. Okay, I want you to know this. Gastrin is a hormone that is released in the stomach, not only when you eat, but when you even think about food, and sometimes after you're done eating. But realize the hormone gastrin is going to cause the release of gastric juice, which is all that hydrochloric acid and mucus and everything. So, and again, that's dominated by the parasympathetic nervous system there in step 1. I would like you to know these 3 phases on the table. Very briefly, the cephalic phase. I can think about food, I can see it, I can smell it, and I'll start secreting gastric juice because gastrin is released. Once the food hits the stomach, that's called the gastric phase, the second on the table, and that will cause more gastric juice to be secreted. And then finally, the intestinal phase, once that food moves into the small intestine where it ultimately is digested, I look at this like a rinse cycle. right? You're rinsing out the remaining contents of the stomach, pushing them on into the intestine. So just know the difference in those. Let's go on. Let's go on. I'm going to get kind of to the point here. The pancreas. Last semester we said this has a dual function of both endocrine gland and exocrine gland. The endocrine function is the secretion of insulin and glucagon in the handling of blood sugar. The exocrine function is in this chapter. It's digestion. It's going to release pancreatic juice into the duodenum of the stomach. And we're going to talk about that here in just a little bit. You can see the intimacy of the pancreas, the gallbladder, and the duodenum here, the first part of the small intestine. And these ductwork emptying into that. As I say to most classes, I don't get to say it to you guys in person because you're online, we are not even scratching the surface of anatomy and physiology. We are in a 139 level class, but when you have to learn all this and the interrelations, and it's not warranted in this class. All right, let's go on here. Pancreatic juice. There's a couple terms here I want you to know, just the basic term. What is in that pancreatic juice that is actually causing digestion to happen? In a minute, we're going to say the small intestine is the primary site of chemical digestion, not the stomach. But I want you to know the term amylase, lipase, nucleases, and proteases. And what each one of those is designed to break down. In 137, we talked about the 4 organic compounds: carbs, lipids, proteins, nucleic acids. So amylase breaks down carbs, simple sugars. Lipase, fats. Proteases, proteins, and nucleases, nucleic acids. That is what, we're not giving you specific names of these because there are a bunch of them. Just know the basics of what they're doing, each one. And that's what is in pancreatic juice. Okay, how do we release pancreatic juice? This is a little flow chart. I don't necessarily like them, but concentrate here on number 2, secretin. Highlight that word, secretin. That is an enzyme that is released, or actually it's a hormone, that is released once the acidic chyme, and we haven't defined that term, chyme, this is what is coming out of the stomach. This is a semi-solid food from the mixing, but it is still not digested. That is very acidic because of the hydrochloric acid that the gastric pit cells are releasing. So when it notices that acidity, it detects that in the small intestine, it releases secretin, the hormone secretin, which will tell the pancreas, gets in the bloodstream and stimulate the pancreas to release pancreatic juice. So that's what stimulates the pancreas to release pancreatic juice. That is secretin. While I'm on this slide, there's another slide like this later on. Cholecystokinin. Now, most likely if you eat a pizza, full of pepperoni and cheese and fat, that chyme coming in is going to be very fatty in nature. It should also be acidic, but let's concentrate on the fat content. Fatty chyme, high fat content, will stimulate the release of this, cholecystokinin, just like secretin. It gets in the bloodstream, but in this case, it's going to stimulate the gallbladder to release bile, and we'll talk about that in a minute. Bile has salts in it to emulsify fats, which means it breaks it into little droplets to be digested easier. So know that, secretin, cholecystokinin, and what stimulates either secretin release or cholecystokinin. All right, the liver. The liver is the largest internal organ. Again, we're not learning all these structures in anatomy. We're not learning it in the lab, just too much for this class. However, I want to get up here to liver functions. Look at the list of liver functions. Memorize them all. I'm kidding. I want you to know the first 2 and the last 2. Let's make it easy. First 2 on the list, last 2 on the list. Produces glycogen from glucose. We probably didn't give you this in the online version, but in my in-person classes we talk about glycogen being made from glucose after you eat. After you eat, glucose is high, body releases insulin. Insulin causes the conversion of glucose to glycogen by joining monosaccharides together like this, one after the other, until we have the polysaccharide glycogen. That's stored in your liver and skeletal muscle until you need energy. Then it will do the second function. It will release glucagon, which will break down glycogen into glucose. It'll start peeling off glucose as we need. That is also a function of the liver, to actually do that conversion. The pancreas releases those hormones, the liver does the work. The liver is a very important organ. Let's get to the last two. I'm not minimizing any of these functions, I just don't want you to have to learn 20 functions, it's ridiculous. Phagocytosis of worn out red blood cells. This is a function that the spleen also has. This is why people can lose their spleen in an accident, they have to remove it, and the liver will carry on that function. However, I would not want to necessarily live without a spleen. Your immunity is going to be compromised. But the liver does that. And finally, it removes toxins like alcohol, drugs. You know, people talk about over-the-counter drugs aren't harmful. Some of you may remember the Tylenol scare a few years back where they just took Tylenol off the shelf because everybody's saying, oh, Tylenol is destroying people's liver. Yeah, when you take 5 a day, 4 times a day, because it's going to destroy the liver. Anything like that is abused can destroy your liver. So it's a detox organ. Those are the 4 functions. Let's go on. I got to get through this. This is an hour and a half long. The gallbladder does not produce bile. It stores it. The liver produces bile. The gallbladder simply releases it. This is the little flow chart about cholecystokinin up here that I just talked about. You can look at that. What is in the bile? Salts. Salts that emulsify. It's like they're scrubbing the fat. It's like a detergent. It breaks it down, decreases the surface tension, breaks it apart so it can be digested easier. Small intestine. This is the primary site of chemical digestion. It is not the stomach. That's primarily what I want you to know there. We'll learn the parts in the lab review. There's your simple columnar epithelium with the microvilli on it. This term plicae circularis would be a good thing to know. This is the alimentary canal. Let's just say this is the part of the small intestine opened up. Can you see these like corkscrew? It's almost like the threads of a screw. And that is caused from all those villi and microvilli that are wrapped around that 360 degrees. So you've got all this surface area to absorb food in kind of a small tube. But it swirls there and probably aids in it moving through. But those circular folds are called plicae circularis. Skip it. Skip it. Let's get to the large intestine here. Again, there is the term peristalsis on that, finally. Large intestine, primary function is water absorption. Water absorption, production of feces. This is the last place you can absorb water. Many people that have a headache, if you drink 2 bottles of water, it's gone because you have a constipation headache. If you're constipated, you're not having a bowel movement, there's no water being absorbed, you're going to get a headache possibly. But know that, water reabsorption. It has basically no digestive function. But we also produce feces. Here's some of the functions. Absorbs water and electrolytes, forms feces. That's primarily it. Feces, all I'm going to say is you should look what's coming out of you. You should glance in the commode, make sure your feces isn't ribbon-shaped. It doesn't have bright red blood on it. It doesn't look like tar. This can tell you a lot about your health. I don't have time to go into that. I'm not going to test you on it. But at least take a look at what's coming out of it. The end. That is the review for lecture exam 1. Again, I'm going to post a separate video about the blood flow through the heart to go along with chapter 15, which will have a lot of good information on it. So this is it. We're going to cut it off because this will be in 2 parts already. It's going to take forever to upload.